Acute myeloid leukemia is notorious for its rapid and lethal incidence of relapse, occurring sometimes as quickly as 3 months after remission is achieved. The goal of my thesis work is to understand how the neighboring non-cancerous cells of the bone marrow contribute to AML relapse. Now, in order to understand cancer, we must understand where it comes from. Leukemia is the cancer of the blood-forming stem cells that live in your bone marrow. Now, normally, these cells exist in a balance that's maintained through reciprocal communication with the stromal or supportive cells of the bone marrow. And aside from supporting the stem cells, these stromal cells also make up the architecture of the bone, synthesizing the fat, cartilage, and calcified bone of the organ. So leukemia occurs when mutations in the blood-forming stem cells prevents them from maturing into blood and instead causes them to rapidly and uncontrollably divide. These leukemic cells are also signaling to the stromal cells, and as the cancer grows, so does its influence, until it convinces the stromal cells to stop taking care of the normal stem cells and instead support, nurture, and protect the leukemia. This remodeling of stromal behavior reduces the effectiveness of chemotherapy. But what we have found is that even during remission, when there is no detectable AML whatsoever, the stromal cells still behave as if the AML is there. Somehow this behavioral remodeling gets locked in. And I believe this is why relapsed AML is so aggressive, because when the cancer inevitably returns, it finds itself in an environment in which it can flourish. So in order to understand how AML accomplishes this stromal reinforcement, I sequenced the DNA of stromal cells from mice with leukemia and compared it to control mice. But rather than looking for mutations in the DNA sequence, we instead looked for these chemical, stable, heritable changes to the DNA. These are called epigenetic modifications. And indeed, AML is able to epigenetically modify the DNA of stromal cells. So I like to think of this as stains left on the DNA from that initial signaling from the leukemia, and this is what locks in that behavior, keeping the bone marrow primed for relapse. Now this seems pretty sinister, right? We have a cancer brainwashing normal cells in the body. However, we know that these epigenetic stains are reversible, and that this can be accomplished pharmacologically. So I believe that by understanding the signaling mechanisms and the subsequent epigenetic changes in stromal cells, we'll be able to develop stromal-targeted adjuvant therapies with the goal of increasing the efficacy of chemotherapy and reducing the rate of relapse. Thank you for your attention, and I hope I convinced you that AML is indeed bad to the bone.